I gave a letter to the postman. He put it in his sack. Bright and early next morning, he brought my letter back. She wrote upon it, return sent. And this son, no such number, no such song. We had one. Alright, I'm sorry, but my letter keeps coming back. So then I dropped it in the mailbox as in a special deed. Bright and early next morning, it came right back to me. She wrote it on it. Then send it.
And that makes me know.
Address.
Okay, that's a big line.
Such person.
So you take on that results.
This time I'm going to take it and put it right in her hand. And if it comes back the very next day, then I'll understand. Return, send it, address. No such number.
This stays here.
No such zone. Return, send it. Return, send it. Don't want a four-leaf clover. Don't want an old horseshoe. Want your kiss, 'cause I just can't miss with a good Lord charm like you. Come on and be my good love charm. Hey, sweet. I want a good love charm, man. To hold, to now.
Yeah. You want to spend the version there?
Don't silver dollar.
We're going to start in 1 minute if people can kind of get seated. Okay, so our next speaker for the immunology section is Sophie Bockenberg from St. Jude and CEIRR. And please continue to post questions for those folks who are online, and we'll have the moderated session after. We'll try to take questions as time allows and audio as well.
I found a lucky penny. I tossed it across the bay.
Great, so everyone's still going back in from tea. Press start. So I wanted to talk about the priming conditions for broadly reactive SARS-CoV antibodies, and this was a close collaboration with Malik Peres. I'm now based at the University of Melbourne, but I was at Hong Kong UPasteur previously for this work. So the University of Hong Kong's had a really long-standing interest in emerging infectious diseases since the H5N1 outbreak in 1997 really prepared Hong Kong to then respond to other emerging infectious diseases. So the H5N1 outbreak then led the 2003 SARS outbreak to have a faster and more rapid diagnostic reaction in Hong Kong, where there was a cluster of cases after importation from China. And then in 2009, the swine influenza pandemic reassortant virus was identified also from the sequencing surveillance work long term from Hong Kong. And then that also led to, in 2012, when the Middle Eastern respiratory virus emerged, having serosurveillance studies established by Mark Pearce, we've been able to look at the endemicity of this virus across Western Africa. And then in 2013, when the H7N9 virus came out, Hong Kong was also prepared to respond to being a close neighbour of China. So all of these viruses meant that in Hong Kong, we've been able to be equipped to respond to these emerging viral threats and really quite strong virology occurring in Hong Kong. And because of the cluster of cases of SARS in 2003 in Hong Kong, we also had access then to SARS-CoV-1 infected donors, which I'll talk about later in this study. So because of the 2003 SARS outbreaks that occurred in Hong Kong, we were also really quite fast to respond to the emergence of the SARS-CoV-2 virus. So early in 2020, in January, schools locked down, and there was a lot of public health measures really quite rapidly introduced in Hong Kong. So with a population of 7 million people, we only had about 12,000 cases over the first 2 years of the pandemic. But recently, at the start of 2022, we've had a BA2 Omicron infection wave, which infected up to 60% of the Hong Kong population. So this means that a lot of the work that I'm going to present today comes from an infection-naive population, because a lot of our samples were collected either during the first year of the pandemic, when the ancestral virus caused the majority of our infections, or during the second year with vaccine introduction, also in that infection-naive population. So we also are going to talk today about some Omicron breakthrough infection cases that were collected earlier this year. But overall, Hong Kong had a really rapid response to the COVID-19 pandemic, which meant that there was really limited cases. So 12,000 cases amongst such a large population initially meant that we had quite effective control of the virus. So in March 2021, in Hong Kong, the vaccination programs were introduced. We had 2 different available vaccines. So this included the CoronaVac whole virion inactivated vaccine. So this is an alum adjuvanted vaccine, and adults and adolescents are getting an equivalent dose. It's relatively thermal stable, which means that it's had a really wide use around the world. So inactivated vaccines actually represent the majority of vaccine doses given worldwide. And initially this vaccine was recommended for 2 doses, 28 days apart. And the other vaccine that we had available in Hong Kong was the BioNTech Pfizer mRNA spike nucleocapsid spike nanoparticle vaccine. So this was there's a different dose range depending on the age group. It's got more lower thermostability, so you have to have that cold chain network. In the initial dosing is 21 days apart compared to CoronaVac being 28 days apart. So these 2 different vaccines have substantially different titers of the neutralizing antibody response, with a 10-fold difference, and CoronaVac being on the lower spectrum of both vaccine efficacy and in neutralizing antibody titers compared to BioNTech. So BioNTech has about a 96% vaccine efficacy against symptomatic disease, whilst CoronaVac is about 50%. And when you actually look at the induction of neutralizing antibody responses for these 2 vaccines, based on the absolute magnitude of the response being substantially lower in CoronaVac, there's also faster waning. So this has led to the update of the CoronaVac vaccine schedule, now being recommended for 3 doses. So we also compared these 2 vaccines, not just for the neutralizing antibody responses, also the FC-mediated functions. So we found that in red, the CoronaVac versus blue BioNTech vaccine responses had substantial rises in ADCC-type binding antibodies for the spike, and these were significantly lower in CoronaVac compared to BioNTech, and BioNTech also outperformed convalescent patients. And the avidity of these responses, even once you adjust for the magnitude, is substantially lower in the CoronaVac vaccinations. So we actually look at the neutralizing antibody responses induced by the 3rd dose vaccination for these vaccines, which then became introduced due to the waning of those responses. We said that the BioNTech has good neutralization against the initial virus, and then the Omicron variants that then started to circulate, especially the BA2 virus, which then predominated during the outbreak earlier this year. Whilst CoronaVac vaccinees that then had the BioNTech vaccine had similar profiles compared to 3-dose BioNTech, CoronaVac alone didn't actually generate protective titers in this threshold band here, where we take a dilution of 1 in 25.6 as giving a 50% protection against these viruses. So this led us to actually say that if you had CoronaVac, and then BioNTech, you could have good neutralization against the circulating viruses, but there was still diminished responses against the newer Omicron variants BA4 and 5. So considering that BA4 and 5 have similar spike proteins but different internal proteins, we can see that BA5 has further escape than BA4 in these neutralizing assays. So when you actually consider the diversity of SARS coronaviruses and the families that they come from, the SARS-CoV-2 is part of the beta family of SARS coronaviruses, and it has many different cousins. So if you actually look at the phylogenetic tree of related sarbecoviruses from different species and the conservation within the beta family of the receptor binding domain compared to the ancestral protein, we see that those that are in the SARS-1 clade, so like the 2003 virus, they have about a 75% conservation of the RBD, whilst in the more closely related pangolin and bat viruses of SARS-CoV-2, they're 88 to 90% conserved, whilst the variants of concern such as alpha, beta, and up to Omicron having increasing similarity to the ancestral virus. Omicron's at about 93% conserved compared to the vaccine type ancestral strain. So we're going to keep this in mind when we look at those cross-reactive antibody responses further down. So the SARS-CoV-2 viruses continue to evolve. We need to have updated variant-specific and combined mosaic vaccines which are currently being developed, and also be able to address future threats from coronavirus family viruses. So what conditions will enable us to have broadly reactive antibody responses against current viruses that are circulating, including its variants and its more distantly related cousins? So it's quite useful if we can identify these conditions to help design which vaccine regimes will generate these responses, and also this can lead then to the isolation of monoclonal antibodies for therapeutics. So we collaborated with Lin-Fu Wang and Xihua Tan from Singapore that initially developed a surrogate virus neutralizing assay where they're using a ACE2 decoy, which can then block the interaction with the RBD protein in the presence of neutralizing antibodies. And so considering that SARS viruses might also have different host specificity and cell types that they'll replicate in, it's quite difficult to have a neutralization assay across all these different viruses. So this multiplex RBD type binding assay is really quite useful to equalize and compare lots of different SARS-CoV viruses. So Lin-Fu initially described in the NEJM paper an 11-plex panel. He's further extended this up to 16 viruses that we'll just use today. And in this initial paper, they actually found that SARS-CoV-1 recovered patients that were then vaccinated with BioNTech had these broadly neutralizing antibodies against both clades of SARS viruses. So they really maximized their responses against any viruses that we could throw at them. So today we're going to talk about a number of different immune conditions in this 16-plex type multiplex assay. So firstly, we look at the magnitude of antibody responses with time post-infection. We compare those two different vaccine platforms that I first introduced, and then in those different vaccines, if there was prior infection with SARS-CoV-2, or if there was prior infection with SARS-CoV-1, and 3 different types of vaccine platforms, including the BioNTech, the CoronaVac vaccine, and a similar vaccine that's actually been more widely used in China, so Sinopharm. And this is also a whole virion inactivated vaccine. We've also then looked with a 3rd dose booster vaccination in a randomized control trial by Nancy Leung and Ben Cowling, where they've had homologous primers with CoronaVac or homologous BioNTech, and then their 3rd dose was randomized for CoronaVac or BioNTech in either group, with pre-post vaccination. And I apologize, but my slides have cut off. And we also had Omicron breakthrough infection where there was people that had a BioNTech vaccination and then an Omicron infection, or a Coronavac vaccination and then an Omicron infection, or also in a vaccine-naive population, so that we can actually look at the true immunogenicity of Omicron itself. And our background controls was a lot of pre-pandemic serum, so you could actually see the relative affinity for the different RBD as well. So apologies, I'm missing all of the data points on here as well and their colouring, but the main message is that between using a bead-based approach and a plate-based approach versus the gold standard PRNT, we saw really good correlation. So this allowed us to then move our assay into that 16-plex. However, if you went from a plate-based approach compared to a bead-based approach, there was a 12.5 times higher signal coming from the plate type neutralization assay. So you just sort of shift your signal upwards. And the reason this matters is because there were some RBD proteins that sort of misbehaved within the assay. So the original beta and delta plate versus bead assays really well correlated with each other, whereas Omicron, due to the number of mutations it had within it, we weren't able to use the bead-based approach. So all of the results I'm going to present today actually then come from the plate-based approach, which had a much better correlation with PRNT results. So my graph is really quite tiny, and again, maybe changed with the formatting. But what we wanted to show here was that pre-post vaccination, in the larger blue bars, the BioNTech vaccine really had SARS-CoV-2 specific antibody responses in this first clade, and the clade 1, so those SARS-CoV-1 type viruses are in the grey boxes throughout. So vaccination alone didn't reach the clade 1 and really only generated its own clade antibodies. So BioNTech boosted responses against 10 out of 16 RBD proteins, whilst Coronavac boosted them against 8 out of 16, but the magnitude of this was significantly reduced in 10 out of 16 proteins. So we have different vaccine conditions here, where we have prior SARS-CoV-2 infection in those two different vaccine platforms, or SARS-CoV-1 with the two different vaccine platforms, or we had collaborators Jinsong Zhao from Guangzhou, where they had the Sinopharm vaccination, and those responses were then seen with time. So overall, we saw that BioNTech vaccination with prior SARS-CoV-2 really only elicited the clade 2 antibody responses, and the Coronavac did have boosted responses, but the magnitude of this was significantly lower at 9 proteins. So it was underperforming compared to BioNTech, despite prior infection. Whilst really only that SARS-CoV-1 infection 18 years prior reached that clade 1 antibody breadth. And that was seen across all the vaccine platforms, but our sample size was much lower for the inactivated vaccines. So just to summarise that together as a heat map view, we could see that the BioNTech had a much larger magnitude response compared to Coronavac, and that prior infection with both SARS-CoV-2 and SARS-CoV-1 with the BioNTech vaccination really increased the magnitude of that response, but it was only with the prior SARS-1 that you could reach that clade 1 type antibody breadth. So it really showed that the mRNA vaccines had an advantage, and that the prior infection with a more distantly related clade virus increased the breadth of responses. To bring back to that randomized control trial of the third dose vaccination, we could see that if you had 3 doses of Coronavac, in the first bars across these graphs, the responses were really quite low and nearly below the detection of threshold of detection across all the proteins. Whilst if you were Coronavac primed but had a BioNTech booster, you could really start to bring those responses back up across the clade 1 viruses with only minor boosting into the clade 2. Whilst if there was a Coronavac vaccination in those that had been primed with BioNTech, there was no further boosting in the antibody responses to any of the RBD proteins, and the third dose of BioNTech gave us the widest breadth to 14 out of 16 RBD proteins. However, when you actually consider the mRNA advantage in that third booster dose antibody breadth, when we actually looked at the T-cell responses in this same cohort, although all my data points are missing, you'll have to trust us that the T-cell responses were boosted in those that had Coronavac priming, irrespective if you had a Coronavac boost or a BioNTech boost. So they had a day 7 T cell response that was coming up at their post third dose vaccination. Whilst those that were BioNTech vaccinated initially and then boosted, there was no T cell rises at this third dose vaccination. So it meant that you have an early rise in your T cell responses from your initial priming, which was really quite useful. So you have to consider then the yin and yang of your antibodies and T cell responses of your boost versus your prime. So we also looked at the Omicron infection breakthroughs in vaccinated individuals, where people that had BioNTech initially then had this antibody breadth to the SARS-CoV-2 clade. Out of 16 out of 18 when they had boosting post-infection. Whilst Coronavac, when they became infected with Omicron, had boosting in a similar clade to only 4 out of 16 RBDs. But in this vaccine-naive population where there's no prior vaccination, Omicron itself really was poorly immunogenic, not establishing boosted antibody responses, and only really to one small variant of concern, and not even Omicron itself. So this was then also conferred with the neutralizing antibodies against these viruses in the authentic PRNT assay by Malik's group, where you could see that Omicron itself really had poor antibody boosting against the different Omicron variants and its cousins. So overall, if we actually compile that data together and you compress the antigenic breadth, so if you consider that how many different RBDs you're binding, so we talk about those RBD cross-reactivity, as well as then the magnitude of the response, so how potent those antibodies are. And you map them out, you can see that with increasing breadth and increasing potency, you sort of reach this upper right quadrant, where those that had prior SARS-CoV-2 infection or that third dose BioNTech type boosting had the largest magnitude and broad antibody responses to the SARS-CoV-2 viruses, and then widening that panel out to include the distantly related SARS-1 clade. It was really only prior SARS-1 infection that gave you those broadly responses as well. So overall, I'd probably like to see this type of experiments then also looked in MERS patients that were vaccinated. So is the advantage in the clade that these SARS-1 viruses come from, or is it the long-term memory? And I think that really help us to define how do you get those broadly reactive antibodies of where you need to actually prime from, what's the sweet spot of antigenic distance for pan-cytoviruses. And I think that's really important for the vaccines we're starting to come through, especially as the Caltech 8 mosaic vaccine is being developed. So thanks to the many collaborators, especially Malik, who helped coordinate the different cohorts, my team that compiled this data and performed the experiments, collaborators for the clinical samples, and Linfer and his team for providing all of these different RBD assay proteins. Sorry if I went over time.
We'll end up skipping questions for this, but we'll move on to our next speaker. Our next speaker is Kuan-Fu, also from St. Jude's CEIRR.
Okay, good morning to everyone. Very happy to be here again, face to face. So I'm going to talk about something that we have learned following a cohort of Chaga disease patients for so many years and what happened after the pandemic with SARS-CoV-2 infection. So briefly, I'm going to describe a little background about the mechanism by which these two pathogens, the parasites and SARS-CoV-2, affect the heart. And then I'm going to describe a cohort of 1,350 patients that we have been following with Chaga disease for more than 22 years and what happened after the infection with SARS-CoV-2. So as we all know, in many of infectious diseases, the clinical manifestations vary from mild disease to severe disease, and it happens the same with COVID. And also, we already know that COVID-19 increased the risk for cardiovascular disease, not only high ischemic disease, but also non-high ischemic disease. So there are several mechanisms by which COVID damage the heart. I described some of them. The cytokine release syndrome, there are many pro-inflammatory cytokines. Also, it can have some acute coronary syndrome mediated by systemic inflammation, and also direct myocardial damage, especially interaction between the virus and some receptors in the tissues. And there are other possible mechanisms with some electrolytes, especially in patients with comorbidities. Okay, and there are also several immunogenetic factors. First, people start studying some host genetics variants, protein expressions, and characteristics of viral receptors. And then also looking association between HLA, alleles, and COVID-19 infection and severity, other immune responses genes, especially related with attenuation or suppression of some cytokines, and coexisting comorbidities also, in this complex interaction between lifestyle, environment, viral strains and variants, and also some variants of genomics of the host. Now a little background about Chagas disease that maybe is new for most of you. This is a parasite, intracellular parasites in the mammals that is transmitted by, it's a vector-borne transmission disease, especially with contamination of the feces of the kissing bags, but it also can be transmitted by contamination of the food, blood transfusion, organ transplantation, of accidents in the lab sometimes. And also when pregnant women get infected, the newborn can be infected. So Chagas disease is endemic in 21 countries in Latin America and even in southern United States, affecting more than 8 million people, 30,000 new cases per year, 10,000 deaths per year, and is the main cause of non-ischemic heart disease. It's becoming a public health problem not only in endemic countries, but also now in non-endemic countries because of migration, because the disease is silent most of the life of the people, and they migrate with the parasite inside their tissues. So the complexity of the natural history, we saw also in other infectious diseases, it doesn't matter which way you get infected, by vector-borne, congenital transmission, oral transmission. You can develop some symptoms in the acute phase, but then you will start getting the chronic phase when the parasite remains in the tissue, and sometimes you don't have symptoms until then, after some years, several years, you can develop heart disease. So in most of the studies, the percentage that people will develop disease after infection, it varies from like 30%, 40% for the chronic stage of the disease. So most people will get infected and remain with the parasite and never develop disease. But still 30% for a disease that is very frequent is very important. The mechanism by which the parasite infection can cause damage in the heart is pretty much the same like so many viruses. So there are different mechanisms like direct damage of the tissue, especially because of the form of the parasite, that they can produce some toxins in the tissue. Also by a specific immunity of the parasite, especially Th1 response, CD8 and CD4, and also non-specific immunity, innate immunity that can damage also the tissues, some kinds microvascular damage in the endothelial cells, and also this parasite can induce autoimmunity because there are some antigens very similar to some antigens in the cardiomyocytes. So, but still we remain with the same question, why some people that get infected never get sick or never develop disease, and others will develop disease. In the past, we were working a lot looking the answers, analyzing the strains of the parasites. But we found out that it's more important the immune responses of the host. So in the immunogenetics of Chagas disease, we have found a lot of MHC class 3 regions, the TNF, for example, LTA genes, that are related with susceptibility to infection and also chronic Chagas cardiopathy. And there is also a variety of HLA alleles associated with Chagas disease in the chronic stage. Also, in cytokines and chemokines, we have found IL-17, IL-18, interferon gamma that are related also, and also some receptors for chemokines associated mainly with chronic forms of the disease. There are also other genes in toll-like receptors that are related especially to natural killers and CD8 in the progression of the heart disease. But now, when we're going to try to study genetics of human populations, it's very complex, especially when we go to Hispanic, Latino population, because of a lot of mixtures, the genetic diversity increased population heterogeneity and may challenge the characterization of disease in different Hispanic Latino populations. In this slide, you will see in the right that, for example, Native American population are more pure, and it happens with indigenous people in Colombia. So it facilitates, in other point, how to do integrated genetic epidemiologic studies in order to identify genetic markers in relation with susceptibility to disease. So also these indigenous people have the highest prevalence for Chagas disease, not only in Colombia, but also in other Latin American countries. However, even if they have high prevalence, doesn't mean that they are suffering of the disease. So they maintain high prevalence because they have different scenarios of transmission that cannot be controlled in the rural areas, in the jungle. And they have transmission by vector-borne or also congenital oral transmission. So these pictures were taken 22 years ago when we started working with Chagas disease in Sierra Nevada, Santa Marta. So we became a reference center for study and research of Chagas disease and other emerging infectious diseases. And 1,350 individuals have been diagnosed with Chagas, and we are following them twice a year, serologically and cardiologically, during 20 years already. All patients with Chagas disease were diagnosed with different serological tests and also PCR according to WHO criterion. Also, in order to assess the degree of cardiac involvement, we did portable EKG, ultrasound, and also the clinical examination every 6 months. And we classified as indeterminate A, B1, B2, C, according if they have some alterations just in the exams or also in the ultrasounds or also in the clinical examination. What happened after SARS-CoV-2 came to the indigenous village? It happens in September 2020 when we got the first case in the indigenous village that they were so isolated in the mountains. And thanks to the project, the natural history of SARS-CoV-2, we could do diagnosis in a timely manner. So 274 Chagas disease patients continued their follow-up even during the pandemic because it was very difficult to arrive to those villages, and all those patients could not be enrolled in the protocol of natural history in the southern hemispheres because we cannot follow them according to the characteristics of the protocol. But at least we got better capacities to do diagnosis in the village, even with PCR and also with antigen. So it's very interesting because people were thinking, well, Chagas disease affects the heart. What's going to happen with COVID that also affects the heart? So we were expecting the hypothesis that it was going to be worse and trying to look for the incidence ratios of cardiovascular outcomes in these Chagas disease patients before and after the pandemic. But then when we realized that the indigenous people have less degree of cardiovascular disease, even if they are infected with Chagas. Less than 10% of the indigenous people with Chagas will develop heart disease. So it's like they have some kind of protection against Chagas disease, even they are infected, but they don't get sick. And in the same way, those few people that got already chronic heart Chagas disease, they develop more symptoms when they got SARS-CoV-2 infections, but they didn't need to go to the hospital. So we have got less mortality rate than the patients, the general population. We cannot compare with all indigenous people in Colombia because the lethality rate, mortality in indigenous people is higher than the general population, but in these tribes in Sierra Nevada is 0%. So
most people got infected, but no one gets really sick. So here we see that we make diagnosis with the PCR, the antigens, the IgM, and IgG. We could not have controls with patients of Chagas disease with no COVID, because when COVID arrives to the village, everyone got infected, even they don't get sick. We could saw that on the antibodies, especially IgG, in December. This was just done in December and was the first outbreak that we got in the village. We knew after, at that time we were not talking so many about the variants of the virus, we knew after that it was the new variant that already arrived in the indigenous village, and it caused a lot of problem in Colombia country, but not in the indigenous population. And we have found also very interesting results in Stacy's lab that the neutralizing antibodies, they don't last. Sometimes they disappear even after 1 month. But it doesn't mean necessarily that they are more susceptible to infection or more susceptible to disease. So contrary to the expectations, I mean, we know that the incident radius of cardiovascular disease didn't change after COVID infection, specifically particularly in this indigenous population. And it could be associated with some immunogenetic factors that we have been studying in the past. We have done some studies looking at the MHC genes associated with tick-borne infection and disease, and we found in some studies that monocytes exhibit the increase of HLA-DR expression in healthy indigenous people, very contrary to what happened in very sick people when they develop COVID-19, and you see in some papers already that HLA-DR levels mirror the balance between the inflammatory and immunosuppressive response in COVID-19 patients. So it is possible that immune response mechanisms that predispose to the development of chronic Chagas disease cardiopathy in these people infected with the parasites are similar to some immune mechanisms related with the severity of COVID-19. When we are trying to explore genetic susceptibility in the human population, it's very, very complex because of the difference of alleles and haplotype frequencies and different genes related with immune response. So it's recommended to study population of diverse geographic regions and diverse population groups. And those genetics variants that are common to other diseases, like in this case, one intracellular parasite, may also help in the addressing future research for susceptibility to respiratory viruses infections. It will be very nice to do some genomics and proteomics with this HLA to see what's happening in this network of T cells and B cells with these indigenous people, because as I told you, interesting, even if they decrease in the neutralizing antibodies, the real life is that they are getting infected with influenza virus, they are getting infected with SARS-CoV-2 virus, but they are not getting sick, or at least when they get sick, it's very mild disease. So we need to go more in deep to see what can we learn about that to improving susceptible people. Thanks. Well, I forgot the acknowledgement also in the last slide for the